Holy cow, here it is, another episode of Mr. W Live. I am so happy to be here with all my friends out in AP Biology, Internet land, all of you guys who are the tough ones, the ones who are ready to do the work, the ones who are ready to bear down and go through the tutorials on sciencemusicvideos.com to do the questions on the BIOMANIA app. These are the people who I am proud to be with. These are the people who I am proud to help guide to a 4 or a 5 on the AP Bio exam. But you've got to be prepared to do the thing that many people are not prepared to do, and that is to work. That is to work really hard on question after question until you achieve the kind of mastery that's going to lead you to the experience that you'll have on May 18th, where when you see those questions, you'll be like, hey, I got this, I got this, I got this. So I am just so happy to be here with Caetano and Michi and Suyu and Matthew and Jyoti. It is just thrilling to be here. Let me just take a little peek at something here. Fantastic. Okay, so we're going to do what we do. We're going to start with the Mr. W theme song. Let's go right here, and let's go here, and let's go here. You guys ready for it? Let's do it. Here it comes. I love this. Me on the beach. Mr. W's live on YouTube. Tuesdays and Thursdays, 2 p.m. PST, to help you learn AP Biology. This BIOMANIA system will help you to review. We'll show you how to write your FRQs. Ask Mr. W all your questions, he's live. We'll guide you to a 4 or 5. Oh yeah. That's right, Mr. W live for AP Bio Review, moving towards the big test that's going to happen on May 18th. Very, very exciting times. I'm hoping you guys have learned so much biology. You know, my whole purpose was to provide a cram-free experience where you could walk into the test on May 18th and just go ahead and do it because you know that material so well. And the way to know that material so well is by practicing. And that's what we're going to do here today, though I am going to do some review. Let's look at our agenda for today. We're going to do a little bit of musical review of DNA replication. I've got a song about DNA replication. I have to admit, it's not my favorite song. I've got some songs that I really love. You might be surprised, but one of my favorite songs is I Want a Clone. And it's about reproductive cloning and why it's such a bad idea. But nevertheless, I like the tune, I like the story. I do some nice guitar riffs on that. This musically, I don't know if it works so well, but the animation is golden and the information is golden, and DNA replication is important. Now, of course, what does important mean in this context? Like, for example, does that mean it'll show up on the AP Bio test? It's so hard to tell because it's just 2 questions, 2 FRQs, one on experimental design, one on disrupted systems. Oh, and I got to show you my disrupted systems video. It's kind of like a slow jam. I'm definitely going to do that. In fact, I'm going to do that after I show you the DNA replication video. And in fact, let's see. Razor's asking, is it true that the exam is on a 3 to 5 scale? 1 to 5. 1 to 5. So 5 is just exemplary and 3 is average but passing, and 1 means it ain't so good. So no, you could get, if it were 3 to 5, then it would be everybody would be passing, and they're not going to do that. Let's see if there's any other questions here. Yeah. Yeah. Okay. What I think somebody's talking about is the AP, the folks at the College Board are putting out some really excellent videos, and I want to encourage you to go over there and look at them.
And they're actually doing practice FRQs, and you should do it. It'll be nothing but helpful for you to have that experience. Anyhow, they have all 10 task verbs. I don't think they're going to put all 10 task verbs in a question, but I don't know. My approach is just practice, practice, practice. Practice on every single FRQ that you can get your hands on, and those are the ones from the College Board. Those are the ones from AP Classroom. That's how you're really going to get there. So, wow, Kitano, you are like a super fan. I love that. Okay, so let's go. Let me tell you what else we're gonna do. We're gonna do a musical review of DNA replication. In our last session, folks asked me to do some review of eukaryotic gene regulation. Will that be on the test? I don't know, but I can review that for you. There was also a request to review thermoregulation. And so I'll quickly go over that. We'll do some biology quiz, and then we'll, I don't know, do some other stuff if there's time. But let's go. So we're going to start with, and Razor is saying, like, any idea what the topics the FRQs will likely be on? No, and nobody knows that. And in terms of illustrative examples, very hard to know that. But I'm actually just going to jump over to my website for a sec. So everybody's looking at my website right now. So go here. Go here. And, listen, it's only like 11 days now before the test, so I'm not saying there's necessarily time for this. But if you work through, units 1 through 6 here, you're going to be set. You're going to be more than set. Work through units 1 through 6 here. Work through units 1 through 6 on BIOMANIA. And what that means is that, like, for example, on the FRQs, you set the thing like this. So you unclick the stuff about the immune system and the nervous system and ecology and evolution. You know that the nervous system and the immune system, they weren't really going to be on the test except for as possible illustrative examples. Like, you don't really need to know the details. But if you do this, then you'll get a random assortment of questions from units 1 through 6. So do that. And then do the same thing for the flashcards. In other words, just set it, boom, like this. Oh, I got a couple of good science practice questions on my flashcards. You should look at them. They review experimental design and things like that. So anyhow, and Suya is saying probably anything from units 1 through 6. And that's right. It can be anything from units 1 through 6. So listen, I wish I could tell you. I really wish I could say it's going to be these topics, but it's not. What you really have to do is some comprehensive review. And if you spend a couple of hours a day doing this, and also go to the College Board website, do their released FRQs, your teacher is hopefully sharing with you the FRQs that are on AP Classroom. That's all you can do. You just got to know a ton of biology, and you've got to be ready to be able to handle the data questions. And Michi, how many hours of studying do you recommend if I haven't started? That's a tough question, and I'm sorry that you're in that position. I am really sorry that you're in that position. But all that I can say is every hour that you spend studying will give you a better result than no hours spent studying. And I would say that like 2 hours of studying on my BIOMANIA stuff, and on top of that, doing those College Board FRQs, that's the thing to do. I'm not saying that the videos that are on the, I'm sorry, I have to have better phone etiquette than I have. I've not been careful about turning off my phone. And I hope I didn't just turn something off for you guys. Okay, no, everything is good. Sorry about that. I don't know if you've ever had this experience, but like, let's say you're in your AP biology class and your phone goes off and your teacher gets like really upset with you. Well, that's kind of just what happened with me, but I was the teacher and I just kind of became upset with myself. So anyhow, I am sorry about that. Sorry for that disruption. There's my phone. The only thing you should be using your phone for is BIOMANIA, because you know there's an app. I have an app for BIOMANIA where you can do these same questions on your phone. So, yeah, Flora is saying something pretty smart. Hay is saying that they've been studying for a month now. Anyhow, Michi, just, you know, do what you can do. You know, I kind of set up a whole program so that you wouldn't have to cram. Oh, but you know what you can do? If you look on YouTube, I have a video. How can I find this? I'm going to go to youtube.com. I'm going to go to, how about this? 3 weeks, week plan for the AP. What happened there? AP. What's happening with my P there? AP bio test. Let's see what comes up. Hey, that's me. I'm the second choice. That's pretty good. So you see this? Your 3-week plan for 4 or 5 on the AP bio exam. So if you go here, and look, I know.
Great enemy of writing.
I know that it's not 3 weeks. Like, you don't have 3 weeks anymore. You have a lot less than 3 weeks. You just have a week and a half. But if you go ahead and do this plan, and it's this plan over here, click show more, and then you see this? That's going to be your BIOMANIA learning journal, Michi. Go ahead, set this up, and just accelerate what's in there. And that'll guide you through all the FRQs. I'm wondering if I can show you that somewhere else. But anyhow, everyone is just being so supportive there. Ansaya, Ansuya, these are all great comments. This is all really smart. And Caetano, flashcards and FRQs. I hope, Caetano, that you're talking about my flashcards and FRQs. But, you know, look, if you make up your own flashcards, that's good. My whole purpose was to try and save you the effort connected in doing that. But it's not like I'm digressing. Guys, here's the deal. I'm going to show you a video about DNA replication. Like I said, not my favorite musical work, but it's pretty good. And here's what I want you to do. DNA replication, super important part. You wouldn't be here if it wasn't for DNA replication, but that's true of everything in biology. Everything, pretty much, because we are ultimately biological beings. We're the product of 3.8 or so billion years of evolution. That's kind of how old our DNA is. Like, that molecule's been replicating itself for that long. That's how long matter has been organized into cells in this incredible chapter that's unfolded in our universe. So in any case, here's what I want you to do. In the chat, what I want you to do is, as you watch the song, I want you to put questions about DNA replication, okay? So as you see something, questions about DNA replication, what's happening here, what was happening here, what was happening here. The chorus, incidentally, is actually a quote from Watson and Crick's paper about the structure of DNA. They wrote it in 1953 or 1952. As old as Mr. Wolkenfeld is, I was not alive then. I was not alive in 1953 or 52. I was born later than that. You might want to guess how long ago that was. You might want to put that in the chat. That's not really important. What's important is questions about DNA replication. Let's watch this, all right? Holy cow, look at that weird expression on my face. Okay. Questions about DNA replication in the chat. Put them in there right now, okay? Go.
DNA structure with its faces complementary makes replication easy, but not quite elementary, since they only bonds with T and C with G. Double helix seems to copy naturally, or as Crick and Watson said, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Your first persons if the DNA in one or more places, breaking hydrogen bonds to separate the bases. Each resulting single strand serves as a template, allowing enzymes to replicate new strands with complementary bases that match. And through hydrogen bonds, these bases attach. Each nucleotide now bonds to the next through a sugar phosphate bond they connect. Meselson and Stahl proved in 58 that this is how the double helix replicates. One strand new, the parent strand preserved. In other words, the whole thing is semi-conserved. It is not escaped unnoticed that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Now let's see how replication really goes with blind, mindless enzymes controlling the show. Made more complex by something you can see. Each DNA strand has directionality. 5' to 3 is how the enzymes go. Just refer to the carbons in deoxyribose. So when a new strand is synthesized, nucleotides get added on the 3' side. The process begins with helicase, which opens up the helix at a special place, breaking hydrogen bonds at the origin. A sequence telling helicase where to begin. A replication fork is now composed, where both parent strands have their bases exposed. And to keep the double helix from rewinding, single-strand proteins come in and start binding. No two forks always form when DNA doubles. The whole thing's called a replication bubble. Now it's primase's turn, the next enzyme, to come to the origin at this time. Primase lays down a primer of RNA, complementary to the template DNA, setting the stage for the star of our show. DNA polymerase now set to go. DNA polymerase's job is to add deoxyribonucleotides to a growing strand, but polymerase needs a growing strand in place, which is why initiation is a job of primase. What happens now is simple, it's a replication race as polymerase follows helicase. As the fork opens up, replication proceeds with nucleotides added at incredible speed. It is not escaped unnoticed that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. What we said applies to the leading strand, where replication smooth, continuous and grand. But on the second strand, fork opens 3 to 5, a direction where polymerase can polymerize. So instead of following helicase, polymerase moves away from the fork in place. So replication's lagging and fragmentary, as discovered in 66 by Okazaki. So the lagging DNA is filled with Okazaki fragments and RNA primers, and to clean up this mess, polymerase 1 removes the primer, puts the deoxyribonucleotides in what could be finer. And now the fragments need to be connected so the new DNA can be perfected. Ligase carries out this function with pride, sealing sugar phosphate bonds between nucleotides. It is not escaped unnoticed that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. It is not escaped unnoticed that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material.
Okay, first of all, let me just address some comments that came up in the chat. I'm going to just set the video. That's a good place. Not there, a little further. Ah, that's fantastic. Okay, so first of all, I just want to address a couple of questions. If you're just starting to study right now, it's not a great situation. I'm not gonna kid you, but you know, it's a pandemic. You're stuck at home. You're not going to go out to the movies. Hopefully, unless you're living somewhere where movie theaters are opening, and don't go to them if they're open, because there's a ton of virus that's out in the population. And you know, you're young and you won't get a bad result. You know, you probably won't get very sick if you get the virus, but you can bring it back to the old folks in your life like me. And the whole point is to flatten the curve of spread so that hospitals don't get overwhelmed. So anyhow, the whole point is you ain't going anywhere. So study biology, like study 5 hours a day if you have to. And the way to study is by doing practice FRQs. Do that over and over and over and over again. You have to get ready to do active production of knowledge. Passively taking in stuff, reading your textbook again, highlighting your textbook, that's a waste of time. You've got to be responding to FRQs over and over again. Use the College Board FRQs and use the ones on my website. That is the thing to do. And guys, I am sorry for teachers who, you know, you're saying in the chat let you down, but I want to say something. A lot of teachers had nothing in terms of online. You know, so like the stress involved in having to all of a sudden deliver an online curriculum was incredible for your teachers. So you have to have some empathy. Like I, just because I'm into this and I've been into computer programming and multimedia design and music, I've been working on this on my website for 5 years. For 5 years, and then this pandemic happened and I was kind of ready. And the College Board helped me out by dropping the last 2 units. I have some of ecology I still have to write. But in any case, have compassion for your teachers. Have compassion for everybody. Okay, let's look at some questions. Flora, you are right. You do not need to know the specifics. And again, someone asked me about DNA replication in the last video, which is the last live I did, which is why I showed this video. But you know what you could actually get asked more about? I'm just going to put this in the chat. The Michelson Stahl experiment. Because what Michelson and Stahl did was they proved that DNA replication was indeed semi-conservative, and that was an incredibly important breakthrough. And the basic idea of semi-conservative replication is that each strand acts as a template for the synthesis of a new strand. And you know, when we think about it now and we watch a video like the one I just showed you, it's hard to imagine another way, but it was a true question that was emerging in the late 1950s as scientists were trying to figure out how DNA was replicated. So I would say learn about the Michelson-Stahl experiment, what they did, because that's the kind of thing with experimental design that could show up. It totally could show up. The 5' to 3' end, you know, that relates to the structure of the molecule. You are not going to get a direct question. Like in a multiple choice question, that could have happened. But on this test with the 2 FRQs, you're really not going to know about this. You know, the basic idea is that the helix splits open, DNA polymerase goes and creates a complementary strand. DNA polymerase is an enzyme. Its active site involves, you know, the other strand and a pre-existing nucleotide, which is why DNA polymerase needs to start with a primer. So there's another enzyme called primase that comes in, lays down a primer of RNA, because there's a way in which primase is more, it has more capabilities than DNA polymerase does. It doesn't need to be adding on to an existing strand. It can just come on in anywhere, and it can start laying down new nucleotides. DNA polymerase comes over, and it'll just add new nucleotides. You have the leading strand where replication is continuous. You have the lagging strand where it's discontinuous, fragmentary, made in short fragments. Someone asked in the chat about the difference between DNA polymerase 3 and DNA polymerase 1. DNA polymerase 1 is coming in and sort of doing cleanup. It's sort of after polymerase, after these RNA primers get kicked out, basically an enzyme has to come in and put in the last nucleotide. DNA polymerase 1 does that. DNA polymerase 3 is the main show. It's possible that I'm getting that wrong. Right now I'm feeling a moment of uncertainty. Teachers experience that all the time, but it's really not important. Big picture on DNA polymerase will, and on DNA replication will be fine for you. So, okay, we'll plant stuff, gibberellins, auxins, bulk flow, apical meristems, and phloem. Well, let's say this. No, nothing in there is possibly on the exam. You don't need to know any plant structures at all. So Newtless, don't worry about that. Newtless, again, you know, of course, if I said I don't want to, you know, promote my own stuff, that's not true. I'm promoting my own stuff. I'm promoting my own stuff because it's a fantastic way for you to learn biology. But again, you know, just go here. Look at this list. This is everything that's on the test. If it ain't on this list, it's not going to be on the test. Now, there might be some illustrative examples, like they could throw in something related to signaling in the immune system or something like that. But that doesn't mean you have to go ahead and do the tutorial on the immune system. What you need to do is do the tutorial on cell signaling, and then you'll be fine. And yeah, Jyoti, you are so on top of this stuff. It's totally impressive. Yeah, it's totally true. They've had somewhat detailed questions about DNA replication. Matthew, in terms of knowing stuff about plant cells, what you need to know is plant cells have chloroplasts, plant cells. You need to know like osmosis related to plant cells, photosynthesis. Geez. But as to like specifics, it's so hard to know because of these two questions. So understand photosynthesis. Understand photosynthesis really well. I've got a detailed tutorial about it. I've got a bunch of questions about it on BIOMANIA. That's the thing that you want to be doing. And Suya is saying something that is golden. Could there be questions, in fact, could there be like an experimental design question that focuses on osmosis? I just looked overhead because there's a light on that shouldn't be on. Excuse me for one sec. I'm back. Okay. Yeah, definitely osmosis, water potential, all of that stuff is fair game for the exam. You know, all of that stuff is in the unit about cells. That's unit 2. So yeah, learn that stuff. Learn this stuff. Learn the stuff related to that on BIOMANIA. And Jyoti, you're right. Big picture. Big picture. Okay. So guys, let's do this. I said that in addition to the DNA replication video, we've already gone over questions about DNA replication, and Puneet is asking about RNA, and Roland is saying that he loves my osmosis song. Thank you. I love it too. That gummy bear thing was a big aspiration. Mock exams. You know what you can do? First of all, people help out Michi and like put a URL in the chat. But Michi, just go like on YouTube, go to AP Bio, like put in YouTube AP Bio review, and you'll get to the College Board. And they actually have some timed FRQs. So do those, and then, yeah. But also, if you just go, I'm just going to do it, because if you just go to any search engine, and if you go here, and if you go past, oh, look at that, it came up, AP Bio FRQs. All right, see what I'm Googling there? All right, so AP Bio, past questions, and I'm hitting that. Okay, and this gets you everything from 2018 back. I'm gonna see if I can put that in the chat. Why shouldn't I be able to? Come on. Ah, didn't go. Try it again. I know how to cut and paste. But maybe not. You know why? Because I have a Windows keyboard here. It's driving me insane. Oh, that's why. Yes. There it is. Okay, guys, you see that? That's how you get to those past AP Bio exams. So hopefully, Michi, that's going to help you out. And then there's also a way you can get to the ones for 2019, but I'm forgetting that right now. But look, if you do all of these, like if you do all of those 3 response questions, like open up that document, go through them, you're going to be golden. Like it's going to be so great for you to do that. They've got the scoring guidelines here. What I tell my students is pretend that you're memorizing a speech, like know them that well. And then if you do that with multiple FRQs, it's like how, you know, a good musician will practice their scales, and then when it's time to improvise, they'll just know it. That's what you're setting yourself up to do. But anyhow, let's go on. Oh, just want to tell you, app $6.99, and I'll give you a special discount on my website. I'm going to move out of the way over here. Okay. So anyhow, go to the contact form on Science Music Videos and just send me an email and say access, and I'll give you a discounted price on access to the website. And that's an incredible deal. Like the amount of learning that you'll be able to do once you have access to all the quizzes on Science Music Videos is phenomenal. You know, I built it for my own students. You can use it. Nutless, the legend. In addition to PCR and gel electrophoresis, what biotech topics do we need to know? I think PCR, genetic engineering. You need to know stuff like this. So here, I'm going back to my website, and here I'm going, that's not where I want to be. I want to be here, and I'm going to Unit 6. So it's the last topic in Unit 6, biotechnology, genetic engineering. You see this introduction to genetic engineering? This is about like how genes get inserted into bacteria. I would definitely know that because that's part of a lab that, well, you know, who knows what happened with the pandemic, but I would have said last year, like everybody does the PGLO lab or a lab like that where you take a gene that's in a plasmid and you insert that into a bacterial cell and then you see that bacterial cell express the gene. So definitely, I just want to show you again, AP Bio, AP College Bio, and then scroll down into Unit 6 and then click Biotechnology and Genetic Engineering. And you know, while we're here, I'm about to do a little mini lecture, but you see it says eukaryotic gene regulation. Let's do that. Let's just do some highlights from that right now. Okay, so everybody know, you want access to my website, just shoot me an email. The app, I can't give any discounts beyond what I've given you, $6.99. Go to the Apple Store, go to the Android Store, Google Play, whatever they call it, and you know, you download the app. The download's free, but then you have to unlock the content. So I'm going here, I'm here, and we're going to do a little bit of review. But you know what? I got to play this because it's like my favorite sound. Everybody ready? Just close your eyes. No, don't close your eyes because watch the animation. Animation. Oh no! I hit the wrong thing. Let's try that again.
AP Bio Review, FRQ.
Whenever you feel bad about the fact that you have to do AP Bio FRQs, just
AP Bio Review, FRQ.
is that fun or what? Okay, let's go. All right, so again, weird test coming up. 2 FRQs. Will there be stuff about eukaryotic gene expression on it? I don't know. But here are the main issues. This is sort of a very condensed version of what I teach my own students. Your DNA, if you were to stretch it out in a linear way, there's a phenomenal amount of it. I think every cell has about 2 meters of DNA. And that's easy to remember these days because for social distancing, you're supposed to stay 2 meters away from the closest person who's a stranger whose infection status you don't know about. So that's how much DNA there is in every single one of your cells. And you have trillions of cells. Trillions of cells. So how do you get all that DNA inside each cell? Okay, second thing, all the cells in your body have the same DNA. But obviously you have cells that make up the lens of your eye and, you know, this tissue in your lip. And, you know, you probably don't have one of these, but maybe you do. So like, for example, the cells that produce, you know, the hair on your beard or the veins inside your hands, you know, the muscle tissue that's in your arms, the enzyme producing cells that make up your liver. All those cells have the same array of chromosomes. In fact, they have the same DNA. So why do cells express different genes? And here's a third issue. Over the course of your life, different genes get turned on and off in different cells, and that has to happen at different times. So like, for example, you guys are all teenagers. You recently went through puberty. Your cells received hormones that were put out by your ovaries or your testes, depending on what sex you are. And those cells responded. And like if you're a male, it did things like give you an Adam's apple over here, or it started to give you facial hair. And if you were a female, you started to develop breasts and things like that. So there are all these other changes that happen, secondary sexual characteristics. So this is, you know, stuff that should matter to you. So here's idea number one. In most eukaryotic cells, only a tiny percentage of genes are expressed. So you've got those 2 meters. I can't even make my hands that wide. Of course, that's not 2 meters. 2 meters of DNA inside every cell. A tiny, tiny fraction of that, a tiny number of genes are being expressed. Now, you can see that under a really good microscope in a weird way. Euchromatin is this loosely packaged stuff over here, whereas the dark stuff is called heterochromatin. And that DNA is packaged up in different ways. The heterochromatin is tightly packaged. The euchromatin is very very loosely packaged. Loosely packaged DNA can be accessed by enzymes like RNA polymerase, which can transcribe that DNA into RNA, and then that RNA can be made into protein. All right, so here's the stuff that you need to know at the molecular level. DNA, right, wraps around these proteins that are called histones, all right? And a group of histones is called a nucleosome. So this over here, this is a strand of DNA. This is the famous double helix wrapped around a nucleosome. And the nucleosome is composed of 8 histone proteins, I believe. Okay, and that forms this very dense fiber, and that's really, all you need to know about that. Okay, so DNA, tightly packed, heterochromatin, not expressed, loosely packed, euchromatin. Euchromatin is expressed. Now let's look at this. A is what heterochromatin looks like, and B, like this, is what euchromatin looks like. This would be expressed, this would not be expressed. And when I say expressed, I mean made into protein. Let me stop and look at the chat, and nothing about this, and Giotti, good. Kitano, nice. That was funny, Kitano. Yensio, you can get a 1. Yeah, 1's a very bad score. Don't get a 1. But guys, if you study, you're not going to get a 1. Just work and you won't get a 1. Okay, so now, there's 2 chemical modifications that happen to DNA. One is called acetylation. And basically, here's an acetyl group. I have a movie. Nah, I'm not even going to say that. Acetyl group, it's one of the functional groups, one of the things that get attached to carbon atoms. So these acetyl groups can get attached to the tails of these histone molecules. And when they are attached in that way, it loosens up the DNA so it can be expressed. So acetylation leads to transcription. Okay? That's the basis of euchromatin. Euchromatin can be expressed. All right, there we go. Now, the other thing is methylation. And when DNA itself is methylated, so I think it's cytosine gets acted upon by an enzyme that attaches a methyl group to that over there. And when that happens, the DNA is shut down. So, like for example, I'm going to take my glasses off. See my eyes? All right, so I've got a lens on my eye. It's a protein that's called crystalline. My cells in my eye lenses, the only protein that they're making is crystalline. Everything else is shut down. So everything else would be methylated, but I'm just sort of speculating here. The gene that codes for crystalline in my eye lens, that is acetylated. So I have a little exercise over here, and I need to move myself over to the right for this. And DNA methyltransferases attach blank to blank. Well, methyltransferases, they attach, let's think about that. What would that be? They attach methyltransferases. They must attach, everybody think it, methyl groups. Oops, that didn't work. Oops. Methyl groups to, it says, cytosine. And the result of this is it blocks transcription of DNA. So let's just talk about how acetylation and methylation are different. Acetylation allows transcription. Methylation prevents it. And the word transcription should be here on the screen, and I forgot that, and I'm just going to have to live with that forever as this thing goes live, onto internet land, and there we are. Okay, so I don't know if you're going to have to know any of this stuff, guys. You do have to know this. So in terms of the structure of DNA in eukaryotes is organized into introns and exons. And the way this is drawn is these are not really to scale. Exons are expressed sequences. They're a relatively small amount of your DNA. In between the exons are expressed, easy to remember, are introns. Introns are intervening sequences and they're not expressed. So let's see what I said here. There's all these other control elements that control whether the DNA gets exposed and how it gets exposed. And I just don't think any of that is going to be on the AP tests. So why am I even wasting your time with this? I'm feeling a little bit bad. But I think everybody just was all flipped out by this thing over here. This diagram flips everybody out. And here's the deal. For eukaryotic DNA to be transcribed, a lot of things have to happen. Basically, you got to set things up so that RNA polymerase will actually bind with the promoter and transcribe the gene. And there's a lot of things here, like at 9, you have this thing called the DNA bending protein. These are called mediator proteins, but you can ignore all that. The only thing you really need to focus on is that a lot of things in eukaryotic systems act as what are called transcription factors. And those transcription factors make transcription of eukaryotic genes possible. So, for example, there's no way that there's going to be a diagram where you're going to have to distinguish between this and this, but just know in general, for eukaryotic DNA to be transcribed, you need transcription factors, okay? So, transcription factors bind near on the promoter. They can promote or they can repress transcription of genes, and they allow RNA polymerase to transcribe things. Once you have the transcription factors in place, then you're ready to go. Okay, so why is this important? Because this is ultimately how genes get expressed. And I'm just going to move myself over to here. So for example, how does this DNA get transcribed? Because a hormone bound with a receptor, a cytoplasmic receptor that went into the nucleus, and it acted as a transcription factor so that this DNA is now going to be transcribed, it's going to be expressed, it's going to be made into protein. All right, let's see. Has everyone just been ignoring me or what? Okay, methyl groups, good. And Suya, I'm just going to respond to a couple of things here. Guys, if you're weak on meiosis, then what you do is this. Go here and do my meiosis tutorial. It'll totally clear everything up. And I also have a song about meiosis, which I played last week. It's called Meiosis, and it really teaches you everything you need to know. Newless, you've been paying attention. Is DNA methylation, histone acetylation inherited? Holy cow, that's such a perceptive question. So I'm going to go back one slide. Or maybe 2 slides. Maybe 3 slides. Okay, these patterns of acetylation and methylation can be loosely inherited in a process that's called epigenetics, epigenetic transfer of information. So for example, what you might want to look up is something called the Dutch hunger winter, which is a phenomenal case study of folks in Holland who experienced starvation during World War II, and they wound up passing on epigenetic changes to the next generation, and those are controlled with food metabolism. So rates of obesity and actually the opposite, like not being able to have normal weight gain, I believe, I'm certain about the obesity thing, wound up getting passed on intergenerationally, which is mind-blowing. The whole field is called, I'm going to put it in the chat, it's called epigenetic, I'm just going to call it epigenetics, and that's all you need to know about it. So learn a little bit about epigenetics and you'll be fine. And LOL, Amanda, though, do we have to know everything down to a T or a general idea? Listen, is detail good? Yeah, detail's good. I totally promote learning detail. But, you know, at this point, you're going to do the best you can. And the way that you're going to do that is by learning how to respond to FRQs. And those FRQs might be ones on the BIOMANIA app. I'm going to bring up BIOMANIA here. BIOMANIA phone app. Did it come up? Yeah, but you can't really see it. That was sort of a useless gesture. BIOMANIA phone app. You can do a lot of practice FRQs. I got the same thing going on my website. Just practice, practice, practice. Do we have to memorize terms like kinetochore, spindle, fibers, etc.? Well, you know, here's actually a really weird thing about this year's test. It's open book. It's open book. So I think if you forget something, like you could possibly look it up super fast, I wouldn't spend a lot of time doing that. I would spend a lot of time going through my flashcards and practice FRQs. So that is really the thing that I'm going to recommend. Guys, let's do some quiz questions. Everybody, fingers ready? Fingers ready? I want to see you typing in the chat in response to these questions. Everybody ready? Let's go. Not that, not that, not that, not that. We did this, we did this, we did this. Okay, it's quiz time. All right, everybody ready? Type some answers in the chat. It's about DNA regulation. Just do it. And let's go. Type your answers in the chat. That is where it's at. I want to see those answers coming at me in the chat. That was a really weak rhyme that I just put together. But let's try this right now. Okay, so first question. Densely packed genetic material that is not expressed. Let's see it in the chat. We'll drink some coffee while that happens. Dealing with the asynchronicity of the internet. Waiting. Fantastic! Newtless, the legend, lives, and indeed it is. Did I say lives? I meant Newtless. Look at all of you guys. Just pour it in those answers. Holy cow. I am so lucky to be, I don't know, your internet teacher or something like that. Okay. Heterochromatin. All right, guys. Next question coming in. All right. Functional group that shuts down genes by binding to DNA. What's it called? GOT, heterochromatin is not really transcribed. No, it's the euchromatin that's transcribed. And I'm seeing the answers come on in. I wonder if people who are closer to me, like geographically, have an easier time with this quiz thing. But I'm seeing all the answers come in. Warm in my heart. Fantastic. Yay! You guys are amazing. Okay, let's do another one. And you know, guys, we're just doing this for the joy of biology. There is not going to be a question like this, obviously, on this year's AP Bio exam. But you know, knowing the details, biology is all about details. I love the details of biology. Guys, what is the name of the proteins that are used for DNA packaging in eukaryotes? I'm seeing it come in. Seeing it come in. Want to see some more? Fabulous. Okay, I think we have time to do one more. Did I already ask this question? I did not. Okay, attaching this functional group to histone proteins makes DNA more available for transcription. Excuse me. Holy cow, you guys are amazing. All right, let's see if I got one more in me. Okay, I'm looking for a very, very general word for this. Signaling proteins that turn on gene expression. It's a 2-word answer, and in this diagram, it's number 11. Once en espagnol. Onze en français. You guys are awesome, and indeed it is transcription factors. Guys, I'm very curious about the pattern of responses that are coming in and the speed. And here's what I'd like everybody to do. I'd like everybody to put in, okay, and I just want to give a couple of people some feedback. Emblem. Exons are the pieces of the eukaryotic gene that get expressed. So that's a little bit different here. So an exon is actually DNA. And Winnie, operators are things that are connected more to operons. I've got a tutorial about operons, and I want you to do that. Can transcription factors turn off gene expression as well? Yes. Yes, they can do both. And how do you say transcription factors in Spanish? Factores de transcripción. I don't know. It's probably that. It probably is exactly that. All right, good. Okay, I think we're done with this. We're not going to do thermoregulation now. Sorry. We're going to do that later. Okay, guys, actually what we're going to do is we're going to wrap up. I want to end with a little inspirational note. Don't go away yet. Don't go away yet, because I don't have I'll just be sad if people go away now, because I'm getting ready to say something. And the thing that I'm getting ready to say is that there is plenty of time left. There is plenty of time left. Like, for example, if this were like normal, like the normal world, I would have been teaching my class until just about a week ago, and then I'd just be having my kids review. And that's it. So you have more time, right? Like, I don't know what's going on with your home situation, your internet connection. There's a million things that might make this difficult, and I'm so sorry about that. I mean, you are the first teenagers who have ever had to live through a pandemic since 1918. There's no one else like you alive. You're certainly the first teenagers who have had to study for an AP Bio exam ever in the history of the universe, because this has never happened before. I'm with you, all right? But you see this relationship over here between time and your goal? Your goal is a 4 or 5 on this test. You spend more time, you got a better chance of getting up to that level. You can do this. You just have to lean in. And the way to lean in is this. You got a couple of things. You're going to do College Board questions, all right? So these over here, very, very important. Watch their videos, do their practice tests, and then spend time on BIOMANIA, which you can get online. You can also, through the same thing, sciencemusicvideos.com subscription, get access to my entire curriculum. It's an entire interactive online curriculum. You will learn so much in the hours that you spend working on that. It's normally $19. I'll give you a little discount if you shoot me an email and say, hey, Mr. W, wanted that access. The way you do that is you see where it says contact over here? Just click contact and it says email me, and just send me an email telling me who you are, and I'll give you a special discount. And then $6.99 to unlock all the content on the BIOMANIA app. And it's worth it. Like, you know, you want to be sitting in like that nice, by your couch, and your parents are going to be saying to you, they're going to say, what are you doing? And you're going to say, I'm studying for AP Bio. And you remember that you gave me the $6.99 on Apple to unlock all the content. I'm doing it now. So I'm working to make you proud, mom and dad. That's the thing you say to them. I would never say no to my children if they said something like that to me. So guys, it's Thursday. Please forgive me for missing Tuesday. I had a conflict I couldn't get out of. I promise I'm going to be there for you next Tuesday, next Thursday, and that's going to be it for this series. It'll totally be it, because that's going to be the Thursday before the AP exam. But keep studying, doing these 3 things. Shoot me that email, and that's going to be it. Guys, everybody be well. Social distancing, don't mess around with that. It's a planetary thing, and I'm there for you. So see you next Tuesday. Shoot me an email for the special discounted price, and everybody be well. Mr. W signing off.